Welcome to Mr. Chalk's Revision Tips. In this video, we will look at protein synthesis, transcription and translation. So, if we remember, the genome of an organism is inscribed in DNA or in some viruses RNA. So the portion of the genome that codes for a protein or an RNA is referred to as a gene. So those genes that code for proteins are composed of trinucleotides, so 3 nucleotide codons, each coding for a single amino acid. So if we remember, a gene is the basic physical functional unit of heraldry. Genes are made up of DNA. Some genes act as instructions to make molecules called proteins. However, other genes act on the DNA in other ways. And if a gene becomes damaged or mutated, it can stop the cell from working properly. So, just to recap, a set of 3 nucleotides is called a codon, and a codon codes for a particular amino acid. So, to enable genes to go and code for proteins, the bases A, T, G, and C get together not in pairs but in triples or triplicates. So this is how it works. So each protein is made up of a large number of amino acid molecules. Amino acids are made in the number and order dictated by the number and order of the base triples. Each triplet of bases codes for one particular amino acid. So finally, the amino acid molecules that join together in a long chain sequence to make up a protein molecules, the amino acids come in the same sequence as is dictated by those triple sequences of nucleotides on the DNA. So the first stage then in producing new proteins is transcription. So transcription is a process in which the information in a strand of DNA is copied into a new molecule or messenger RNA. So DNA safely and stability stores its genetic material in the nuclei of cells as a reference template. So the sequence that happens is that the DNA in the gene unzips, so both strands are separated, so one strand can be used as a template. Complementary bases attached to the strand being copied, so C joins to G and so on. Thymine bases, which is not present and a different form to the base called U, joins the in the way that T would have. So this goes and forms a new strand of messenger RNA. So we'll just have a look at this question. So spiders produce a protein thread which is extremely strong compared with man-made fibres in of the same diameter. Explain how genes control the way the protein in the spider's body is made. Right, some key things is that we need to mention the fact that the gene, which is DNA contains bases. These bases code for amino acids in the protein. So amino acids in the correct order will go and make the spider proteins eventually. The next stage is translation. So in molecular biology and genetics, translation is a process in which ribosomes in the cytoplasm or on the endoplasmic reticulum will go and transcribe the RNA, and using the code from the RNA, it will go and make a protein. So the way that this happens is that the messenger RNA attaches to the ribosome, and the ribosome is basically going to read it. The ribosome decodes the messenger RNA in groups of 3, so these base triples or codons, which are complementary bases to transfer RNA. The transfer RNA is specific to an amino acid that it collects and returns to the messenger RNA that's being fed through the ribosome. The amino acids are now lined up in the order or instructions on the messenger RNA. So bonds form between the amino acids and a polypeptide chain is formed. And the polypeptide chain folds and then becomes a shape, forming a specific protein. So we just look at this. So what is the name of the molecule that carries the code from the DNA to the ribosome, either RNA or messenger RNA? If we're describing how the ribosomes help the cell to make enzymes, right, remember enzymes are proteins, so the ribosomes make the proteins using amino acids and in the correct order as determined by the messenger RNA, which has been made using the code on DNA.
Thanks for watching.